Now the entire gene machine was revealed. It begins near the head of an active gene at a short sequence of DNA called a promoter. Once a chemical go-ahead is received, an enzyme called RNA polymerase attaches itself to the promoter. It then travels along the double helix, unwinding and separating the two strands ahead of it, as if opening a zipper. The RNA bases floating nearby now begin to attach to one of the exposed DNA strands, according to the base pairing rules, with U substituting for T. As the enzyme moves forward, it slides over and around these anchored RNA bases, linking them together through stable covalent bonding. An ever-growing tail of messenger RNA emerges from the traveling enzyme until it hits one of 3 different codons that tell it to stop. Transcription is now complete. Then this single strand of RNA escapes from the nucleus and carries its transcribed message to the ribosome, the machine that translates it into a protein. As the 2 pieces of the ribosome clamp together around the messenger, a transfer RNA attaches to the first codon of the message through the base pairing of its complementary anticodon. It carries the first amino acid. Then the next transfer molecule attaches itself to the next codon. The 2 amino acids are joined together through covalent bonding. The ribosome then moves down 3 base pairs, releasing the first transfer RNA minus its amino acid and queuing up the next one. And in similar fashion, the entire protein sequence is assembled. When the stop codon is reached, the ribosome halts and the completed protein is released. It then folds into its unique 3-dimensional form.
Within a very short space of time, I mean from 1953 to, let us say, the summer of 1961. It's only 8 years, and that sort of the whole thing was laid out.
One of the final pieces fell into place at the Eagle Pub, where one day Brenner and Crick were discussing some odd things in the mutations they had created in bacterial viruses.
We were puzzled by all the problems, I mean, of mutagenesis. They didn't fit. And one of the interesting things is always that most people, you know, just look at the science that fits together. You don't learn anything. You have to look at the pieces that don't fit.
Most chemicals that produce mutations seem to substitute one base for another, as predicted by Watson and Crick in 1953. Such substitutions only affect one amino acid, leaving the rest of the protein unchanged. Crick and Brenner had no trouble detecting these mutant proteins and found they still functioned, at least in part. What didn't fit with this pattern were the mutations produced by a particular group of chemicals called acridine dyes, whose proteins seemed to be missing entirely.
In the case of those acridine mutants, you know, we were just worried about substitutions. And were wondering to say, but you know, it was at that Eagle thing that we started to talk about, well, what if you could add bases or delete bases? And this got very exciting.
Picture what happens if a new base is inserted into a codon. Not only does that codon now read differently, so do all the codons that follow, because their bases have shifted position within the framework of triplets. Something similar happens when a base is deleted. That codon and all the ones that follow also become nonsense words, which is why Brenner and Crick were unable to detect any proteins when they used acridine dyes to cause mutations. The success of the RNA Tie Club was a rebuke to their many critics in the scientific establishment. Sidney Brenner fully savored their triumph.
I could remember sort of sometimes saying to people, you know, we will bury you, which was a Khrushchev phrase. We did bury them.